Hello, nerds. Welcome to MedExpert. If you're looking for reliable, evidence-based medical content, you're in the right place. We provide expert insights, health tips, and the latest medical updates. In this video, we'll explore the essential techniques for studying cells. From imaging methods to experimental tools, we'll cover a range of techniques that allow us to look inside cells and understand their functions. So if you've ever wondered how we study the cells that make up all living organisms, stick around. Let's get started. Cell fractionation, differential staining, microdissections, tissue culture, chromatography, electrophoresis, spectrophotometry. Let's dive into each technique in detail, starting with cell fractionation, and explore how it helps us better understand the complexities of cellular biology. Cell fractionation is used to separate a cell organelle and components based on the size and density of the medium. It is beneficial for electron microscopy of cell components. The principle of cell fractionation consists of two steps: homogenization and centrifugation. Homogenization is the formation of a homogeneous mass of cells and grinding of cells in an appropriate medium with specific pH, ionic composition, and temperature. The uniform mixture of cells formed at the end is then processed for centrifugation. In plants, the enzyme pectinase is added to digest middle lamella. This can be done in a blender. This procedure gives rise to a uniform mixture of cells, which is then centrifuged. Centrifugation is the process in which mixtures are separated using centrifugal force generated by spinning in a centrifuge. The mixture is separated based on its size and density under centrifugal force. Centrifugation is done in a centrifuge machine in which tubes are spun at high speed, which exerts centrifugal force on cellular components. Centrifugation can be performed in two ways: density gradient centrifugation and differential centrifugation. Density gradient centrifugation includes the separation of cell components on the basis of sizes and densities in different layers. The upper layer is less dense, while more dense components settle in the lower layer. Differential centrifugation is done on the basis of the size and shape of particles that determine how fast they settle. The components of the cell that settle in the bottom of the tube are called pellets, while the components that remain suspended above in the form of liquid are called supernatants. The supernatant is drawn off after every step in centrifuge again. A series of pellets containing cell organelles of smaller and smaller size can therefore be obtained. The technique used to differentiate transparent cellular structures from each other by applying various color dyes is called staining. This technique is used because most of the biological structures are transparent. There are two types of staining: single staining and double or differential staining. When only one stain or dye is used, it's called single staining. For example, borax carmine is used to stain the nucleus, while when two dyes or stains are used, it's called double or differential staining. For instance, hematoxylin stains the nucleus, while eosin stains the cytoplasm. Microdissection is a technique used to isolate and dissect out specific cells, tissues, or small structures from a larger sample under a microscope. It is used to remove tumors or granules from delicate tissue or cells, like brain, heart, and nerve cells. In this technique, the image is observed on large TV screen or monitor while dissecting. Tissue culture, growth of a cell or a tissue on an artificial medium containing all desired nutrients in sterile conditions, is called tissue culture. Plant tissue culturing is used for production of genetically identical plants, clones. While animal tissue cultures are done to find abnormality in the cell, for instance cancer, chromosomal disorder, etc. Maintaining a sterile environment is critical to prevent contamination from bacteria, fungi, or other microorganisms that can compromise culture results. Cultures are maintained in incubators with controlled temperature of 37 degrees Celsius, humidity, and CO2 levels to mimic the natural physiological conditions of the cells. Chromatography is a technique used to separate various chemical compounds from a mixture. It is commonly applied to separate biological molecules, such as proteins, amino acids, or photosynthetic pigments. There are several types of chromatographic techniques, including gas chromatography, thin layer chromatography, paper chromatography. We'll explore these techniques in more detail in another module. Electrophoresis. Electrophoresis is a technique used to separate charged components of a polymer molecule based on factors like shape, size, molecular weight, and surface charge. That charge-bearing polymer molecules can be RNA, DNA, or protein, etc. In this technique, a gel medium is used to separate fragments under the influence of an electric field. The gel is often placed between glass or plastic plates to create a thick, viscous slab. The two ends of the slabs are placed in separate salt solutions, which are connected to electrodes linked to a power source. Samples are loaded at one end of the slab, and when the voltage is applied, the molecules in the gel move through the electric field. Negatively charged molecules are attracted to the positive pole, while positively charged molecules move toward the negative pole. The relationship between the velocity of the fragment and its size is inversely proportional, meaning smaller fragments travel faster than larger ones. Nucleic acids like DNA and RNA strongly absorb light at approximately 260 nanometers because of the aromatic nucleotide bases they contain, like adenine, guanine, cytosine, thymine, and uracil. This absorption at 260 nanometers is frequently used to measure the concentration of nucleic acids. And that concludes our module on techniques in cell biology. If you enjoyed this lecture and want to see more interesting content, be sure to like and subscribe to our channel.